Good morning, good afternoon, good evening. It's 10:05 now, and we would get started. Welcome everyone to this online hands-on workshop on RxODE. So before we get started, I actually just want to check how many of our participants are here. We actually had close to 500 registrants for this workshop, and we're really excited to have you guys join us. In this workshop today, we have participants from all over the world, from India, South Africa, China, Singapore, Malaysia, and we are really excited to have you all here with us. And thank you, Wenping, for taking on hosting this workshop for us today. So a little bit of introduction before we get started. This workshop is hosted by Project Bhakthani and ISA, and it is sponsored by PharmaPro Consulting and CRC Pharma. So a little bit about your host, Aruna Bhakthani. I am the founder and CEO of PharmaPro Consulting, and I'm also the founder and scientific director of Project Bhakthani. Prior to doing this, I was a director at InClone Lilly and headed the clinical pharmacology and pharmacometrics team there. In my career of 25 years, I've worked for multiple pharmaceutical companies supporting clinical pharmacology and pharmacometrics, and have supported compounds, small molecules, as well as biologics throughout the drug development. So something about Project Bhakthani. We are a nonprofit organization created to support scientists, students, and postdocs from around the world in the field of pharmacy, and our objective is to support scientists in growing their careers by providing them workshops, training programs, and mentoring programs. Now, how can you support us? You can support us by joining hands with us and helping us in creating similar workshops, and definitely taking on being a mentor in the mentoring programs. And so is our motto, which is empowering you to empower others. A couple logistical questions, or rather a couple logistics of how this webinar will go before we progress further, is all our participants are going to be on mute throughout this workshop. However, you can reach out to us via the question and answer box, which should be either on top or the bottom of your screen, or you can also send us an email at dontabhaktani.org@gmail.com. And if any of your colleagues and friends have had issues reaching out to us or have not received the link for this webinar, you can ask them to send us an email to dontabhaktani.org@gmail.com, and we should be able to share the link to join the workshop. If not, we are recording this workshop and we'll be sending you the link to the recordings, as well as the slide deck and the homework assignments of this workshop. So Project Bhakthani has been in existence for a year now. And last year, myself and a colleague of mine, Rakesh Golan from CRC Pharma, we had traveled to India and hosted multiple workshops in many locations and had over 300 participants attend these workshops. We also did travel to China to host the same. And this year, while the world is dealing with the pandemic and there are travel restrictions, we have taken on doing online workshops similar to the one that you are part of. And our intention is to have as many workshops as possible, intending to have at least once a month for the next several months to go. Our next workshop, which is coming up, is on the subject of clinical pharmacology. And the title is How Clinical Pharmacology Shapes the Strategy of Clinical Drug Development from NME to Registration, which is going to be presented by Bhakthani, Senior Director, Handson. And the dates are August 19th and August 26th, very similar to the kind of format that you're currently part of. A few words about our sponsors. One of our sponsors today is CRC Pharma, which is a consulting company specializing in supporting biostats, clinical pharmacology, statistics, clinical data management, clinical PK/PD analysis, pharmacovigilance, and medical writing. CRC Pharma has been in existence over 10 years, and they have supported 250 studies over different aspects of regulatory submissions. And they are your go-to people for non-member ready data sets and any kind of data-related questions. Now, PharmaPro Consulting. PharmaPro Consulting is federally and state certified as a women-owned and minority women-owned small business. We specialize in PK/PD analysis, clinical pharmacology, strategic advice, mathematical modeling, regulatory affairs, and CMC. We have been in existence for 5 years, and we have worked with multiple clients like Genentech, Daiichi Sankyo, Novartis, SK Life Sciences, to name a few. Our team of experts have over 25 years of experience individually and have supported multiple compounds over different phases of drug development. Our experts have experience working in small molecules as well as biologics and vaccines. We have supported the gamut of therapeutic areas. Our experts have experience working with various regulatory agencies. We have supported breakthrough therapy designation, fast track approvals, INDs, NDAs, BLAs, biosimilars, as well as 505(b)(2)s. So we really take pride in saying that our motto is to save our clients time and money. We have used mathematical modeling tools to help our clients reduce their trial sizes by 91%, and also by helping them determine the effective dose of compounds early on in drug development, saving them millions of dollars in costs as well as drug development timelines. Again, a workshop of this magnitude would not have been possible without the support of multiple people. And I would like to take this opportunity to acknowledge our ISAB board, who are also the co-hosts of today's workshop, and our sponsors, CRC Pharma and PharmaPro Consulting, and the interns in Project Bhakthani, Ramya, Krishna, and my board of directors, Rajan, who has been part of this whole initiative. I would also like to acknowledge Ramesh Naidu, who is part of the PharmaPro team, who has been working relentlessly to make sure that today's workshop is conducted seamlessly. Now, without further ado, I would like to introduce our speaker for today, Dr. Wenping Wang. Dr. Wang is a distinguished scientist who has worked in consultancy as well as pharmaceutical companies, including Farsight, Johnson & Johnson, and Novartis. Wenping was awarded the ISOP Innovation Award in 2018 for his exceptional innovation and advocating the discipline of pharmacometrics. He was named a Novartis IVA in 2017, and this award was given to celebrate his exceptional work and contribution in the area of drug development. Besides his essential contribution for several successful NDAs across multiple disciplines, the primary reason for his IVA award was his work in the transformational nonlinear mixed effects modeling software, NLMixR, a free open source package for R. NLMixR builds on Wenping's RxODE package, a tool for simulating PKPD models using ordinary differential equation. Now, without further ado, I welcome Dr. Wang to take on this presentation. Give me 1 second while I stop sharing my screen so Wenping can take this on. Wenping, the floor is yours.
Very good. Thank you so very much. And let me know if you guys can see the screen.
Yes, Wenping, I can see your screen.
Very good. Thanks a lot, Aruna. Thank you for organizing this event. And I thought you're doing a really tremendous service to this ISOP community. And hello, everyone. I hope you and your family are well and safe among this COVID difficulty. So today, on behalf of the NLMixR team, I'd like to give you a brief hands-on intro of simulation with RxODE. And so first, an outline of today's topics. I'll start with some RxODE basics, then proceed to regimen simulation. After that, it is the population simulation with RxODE. And afterwards, I hope we can have some fun with the shiny interface of RxODE. And towards the end, I'd like to introduce some advanced topics. So what is RxODE? RxODE is an open source R package. It is designed for pharmacometrics simulation. This software was initially written by myself and Matt Fiedler. It's available on CRAN, GitHub, and described in the tutorial in CPT-PSP. And there are a lot of online documentations that you can take a look. And after this webinar, we are going to distribute the slides, and you can find the links to those documentations. And if you have done simulation using R, you would know that simulation of differential equations was possible in R if you use DE-solve. But if you use DE-solve, it can be quite slow. And if you have a flexible and complex dosing history, it's virtually impossible to code. So in contrast, RxODE allows fully flexible dosing history. And behind the scene, RxODE is compiled in C, so you have rapid execution time. RxODE has been in the public domain for several years over multiple operating systems. RxODE is considered a stable and a mature software. However, if you want to use RxODE, you will need an external compiler so that the code can be compiled and executed. So just to give you an idea about the workflow of using RxODE to do simulation with dosing regimen. First, you need to load this library RxODE. Afterwards, there is a function, we call that RxODE. Within this function is a series of assignments that you define your differential equations. And this little magic function RxODE will do code generation and compilation for you afterwards. The compiled ODE object on the fly is assigned to an R object. And you need to provide the system parameters that you use when you define your differential equations. And you need to create an R object called EventTable. So this is a place that you specify your dosing regimen and where you want to do the samples. And afterwards, there is a method with the compiled RxODE object. We call that solve. You call this solve method to do simulation. Let me get into the details of RxODE. First, about the syntax. RxODE, I believe, is quite simple and intuitive. Differential equations are specified in series of assignments. Your derivatives with respect to time is coded as d/dt, then in parentheses, the state variable name or compartment name. You can define additional derived variables. If there are certain quantities or variables you used when you define your differential equations, you referenced it, but you had never defined it, then all those referenced undefined variables are automatically assumed to be input parameters. So input parameters must be specified as a named vector. I'm going to get into that a little bit in detail later. The order of the input parameter vector is not important. However, the names of the input parameter vector must be a superset of parameters that you used when you define the differential equations. And optionally, there are additional attributes of compartment be specified. I will touch about this one a little bit later, and towards the end in the advanced section, I'm going to discuss this part in a little bit more details. So let's get into a little bit more details of using RxODE. And what you want to do is you load up this library, RxODE, then you say within this function, RxODE, then in parentheses, in brackets, curly brackets, you define your differential equations. And in this example, I defined 4 compartments. One is called the depot, the other one is called center, yet another one is called the peri, stands for peripheral, and the 4th one is defined as EFF. So you can see these 4 derivatives are a series of assignments. I also defined a couple of derived variables. C2 is equal to the amount of center over V2. And C3 is equal to the amount in peripheral compartment over the volume V3. So here, the V2 and the V3, they are referenced, but never defined. So therefore, RxODE would assume that V2 and V3 are parameters to be defined later. So there is another sentence that's worth your attention. That is, this EFF0 is equal to K in over K out. What this assignment means is the starting value of EFF is equal to this K in over K out. After you define this, so this RxODE, this function will compile the code and produce a binary ODE solver. We call that M1. And there are several parameters we used when defining the differential equations. Now you use a named vector. The so-called named vector actually is just a regular vector, but you say Ka is equal to so and so, clearance is equal to so and so. And next on your to-do list is you need to create an event table. So EventTable is a container that has your dosing information. In this case, it's a single dose. And also, it has the sampling information, where you're going to do your concentration measurement. And after you have the differential equation object M1, your input parameters, your event table, then you call this method solve to do the ODE solving or prediction. Afterwards, there is yet another helper method called plot to plot out different simulated results. And if you do not specify any quantities like what I did here, C2, then by default, it will simulate all the state variables as well as the derived variables. So the important part is to specify the dosing information. And an event table is a container that stores dosing and sampling times in a chronological order. So the dosing part, you can call this function add dosing multiple times. What it does is incrementally add the dosing records. And you can do the same thing with observation times. And we can take a look of this dosing function in details. What you want to do is you need to tell what's the dose amount you want to give, how many doses you want to give, and what is the dosing intervals. By default is equal to 24. And where do you want this dose given to? Default is given to the first compartment. If you have infusion, then you can specify your infusion rate here. And if you want to start your time, your dosing time at different time, then you can specify the shift with this start time. And the method solve is also pretty easy to use. What you want to do is you specify that compiled ODE, RxODE object, and you tell this solve what's the input parameter. Remember, it's a named vector. And also, what's your event table? Event table is the place you specify your dosing information and your observation information. So with that, let me go back to the hands-on part. Here, I would want you to take a look at some examples. And afterwards, Aruna would distribute those examples for you to play and practice. So let me load this object, then I compile this object. Afterwards, I say assign that a name and a vector to an object. I call that theta. So I define a little helper function to do the ODE solving, as well as to plot the simulated C2, which is the drug concentration from the center compartment. So find this. Now, here is the most interesting part in your regimen simulation. For a single dose, you say your dose amount is equal to 10 milligrams, and your number of doses is equal to 1. Then I want to sample every hours from time 0 to hour 24. I run this code. Now you see it gave me the profile of a single dose drug concentration. Next, I want to do multiple dose. Multiple dose actually is pretty simple to do. All you need to do in this case is you say number of doses is equal to the dose number you want to simulate. I want to simulate 5 doses, and with a dosing interval equal to 24 hours. And you do this again. Now you have 5 doses. Next, what I want to do is I want to do a BID for 5 days, then afterwards QD for another 5 days. So the way you do that is first you call at dosing once, just like what we did before, but the dosing interval is equal to 12 hours. Afterwards, you say, I want to do QD. So therefore, my dosing interval now has been changed. Yet another one is, I need to shift the starting dosing time by 5 days. And if you do that, now you have this PID for 5 days, then QD for another 5 days. Next, what I want to do is, I want to do infusion for 5 days, followed by oral dose for another 5 days. For infusion, what you need to do is, there are a couple of things you need to do. One is tell the system that at this time, my dosing events is to the central compartment. Dosing 2 is equal to 2. 2 is corresponding to how you defined your differential equations in this definition. And another thing is you need to specify your dosing rate. So in this case, dosing rate is equal to 5,000. And afterwards, you specify your oral dose, like what we did before. When you do this, now you have 5 days for infusion, followed by oral dose for another 5 days. I work in oncology. Quite often, I need to do regimen simulation. In that case, I need to specify certain chemo, how many days I want to give the patients with this chemo, and how many days I want to give patients some time to recover. And in this case, for instance, I want to do a 2 weeks on, 1 week off simulation. And I want to do it for 4 cycles. What I need to do now is, you noticed, I need to do I need to call this add dosing multiple times with different starting time. If I do this kind of thing, I run this section, I have this 2 weeks on, 1 week off regimen for 4 cycles. Next, I want to explain to you is the population simulation. Population simulation is also quite straightforward with RxODE. If you remember, for single subject simulation, what we did is we create a named vector. So that's a key step, so that we can do a single subject simulation. However, if you want to do a population simulation, what you need to do now is to create a data frame with all the parameters. So let me run this piece of code. Afterwards, I have this object called theta.all. If you take a look at this one, I have 100 sets of parameters. If you have this data frame, the rest is very similar to single subject simulation. You define your event table. Now, if you remember, when we do the single subject simulation, the calling convention is your compiled RxODE object followed by that parameter theta and event table. The population simulation is almost identical to that single subject simulation. You have the compiled object, and then now you replace the previous named vector by a data frame. Then afterwards, it will simulate this 100 subject for you automatically. Actually, the simulation is pretty quick, but when I call this ggplot, it takes longer time for ggplot to do this simulation. So for population simulation, there is yet another one. You know, you may have some clinical trial data, and you have your non-MEM data set. You wish to use the non-MEM data set to do the simulation. The way you do that actually is also very simple. You guys may remember this famous theophylline dataset. So that's initially published by Louis Scheiner when they create a non-MEM for the users. So this is essentially the non-MEM dataset. What you want to do is you can use a utility available from RxODE called as.et, stands for as EventTable. This will translate your non-MEM dataset to a EventTable object. So let me go ahead and run this one. Now afterwards, you can just, okay, let me also generate this 12 subject parameters like what we did before. Now you see my event table is this theophylline data set. Now you can see I have 12 subjects with the event table that's imported from a non-MEM dataset. I noticed we only have 17 minutes left. Aruna, I want to check with you about the logistics. I still have a couple of sections that I can go, but it will be a rush. Another possibility is we move these last two sections to the next week. So what's your suggestion, Aruna?
So Wenping, one is that we have a couple questions which have been asked. We have 3 questions that I would like for us to address. And overall, I would say at the end, I might just need less than 5 minutes. So, so far, we can take on addressing the questions. Rajan, can I request you to please read the questions for Wenping so he can answer them? You should be able to get yourself on mute, Rajan.
Yeah. Yes, Aruna. I am seeing few questions here. I will read out to Wenping. Okay. If possible, can we please discuss dosing simulation for weight normalized regimen, something like mg per kg basis, from adult dose to pediatric dose? That's one question, whether it is possible for such kind of weight normalized dose.
Right. So it is indeed possible. Actually, I am glad. I want to thank the the colleague who raised this question. It's just interesting to see that I myself actually, I have one such example explicitly to talk about how to do weight-based simulation. Let me see. You may notice what I highlighted here. So my suggestion is I give you a short answer. It is possible to do that. And maybe next week, we're going to cover this weight-based simulation in a little bit more details, plus the Shiny interface, as well as the advanced section.
Thanks, Wenping.
Yeah.
Next question is how to add variability.
How to add variability. So that's, again, a very good question. In population pharmacokinetics, you have two sources of variability. So you have the so-called between subject variability, and you have the so-called within subject variability. So in this example, actually I demonstrated the between subject variability. Let me go a little bit slower here. So my clearance, the population median is about 18.6. What I want to do is I want to introduce about 10% between subject variability. And I'm going to assume the between subject variability follows a log normal distribution. So therefore, you can see the coding here. In R, of course, R norm is a function to generate random variables from a normal distribution. So in this case, when I just call this R norm, let me change that to a smaller number so that you guys can see it. When I call this R norm, you noticed I created 10 random numbers follow a normal distribution. If I do this together, I will add some log normal random variability to this central tendency of my clearance. So when you do that, now you see I have 10 clearances from 10 subjects. So this is, let me run this whole thing for you. Now if you take a look, it gave you 10 sets of PK parameters mimicking 10 subjects in the population. After you have do this simulation, let me run this part again. If you take a look, what's coming out of this simulation, let me show this to you here. Now, sim.id is the subject simulated patient ID. So now we have 10 subjects at a different time. Their concentration is denoted as C2. And suppose you want to add some intra subject variability to that. Now what you can do is you can get your C2. So these are the all the simulated patient's drug concentration. Now you can say, I want to add more variability to that. So what you can do is you say R norm, you say you want to generate so many X2, C2, then you say with mean equal to this, with standard error equal to so and so, standard deviation. So this will add the within subject variability to your simulated data. So let me stop here. Aruna?
Yeah, thank you, Wenping. And a few more questions are there. Shall we continue?
Yeah, sure.
Okay. Okay. So this is a question from Du Yang Lee. Can RxODE do the fitting to data? For example, fitting using two-compartment model to estimate PK parameters.
Right. So that's again, that's a very good question. RxODE by itself do not do curve fitting. The curve fitting is from the sister package. We call that NMXR. NMXR can do population model fitting or single subject curve fitting there. You can define your model as the differential equations and do the curve fitting there.
Okay. Another interesting question from Vivian. The model defined as 3 compartments. How does the simulation, that infusion simulation work automatically? Does it recognize the input to the central compartment and skip the dosing or the absorption compartment? When we switch to infusion, how that it recognizes the central compartment and directly get the drug there?
I see. Okay. All right. So this again, that's a very nice question. This actually is related to the behind the scene operation. What it does, Vivian, is let me go back to that example. I'm explain a little bit about the behind the scene operation. There are several key things. One is this dosing 2. So where this dosing event happens. By default is equal to 1. If your infusion rate is equal to now is equal to nothing, then RxODE assumes that it's a bolus dosing. So what it does is, if depends upon where you want to put the dosing to, RxODE will just add certain drug amount to a particular dose, to a particular compartment. Let's say in this case, let's say if my dose equal to 2, behind the scene, RxODE will add some dose to this compartment. However, if it's an infusion, when you have infusion, your dosing rate is equal to a number. And when you do that, RxODE understands that you are going to do a drug infusion, and also use with the dosing tool, then you're going to say this infusion events happens to the second compartment. So what it does is, again, behind the scene, it will have a technical term called forcing function. So there is a forcing function equal to your infusion rate added to this differential equation to mimic your drug infusion. I realize that this is a little rather technical. The magic is these two things. These two things will determine whether it is an infusion event or whether it is a bolus event, and where does this dosing happen to.
So Wenping, in the interest of time, I actually wanted to bring us back to the hands-on workshop piece of today's discussion. Again, I do see that there are several other questions which we have not been able to address yet. I want to assure you that all your questions will be answered. And that is the reason why we have created this as a hands-on workshop for this week and the following week. So the next step from here on is going to be that Wenping has created some hands-on assignments that you can work on. And the way we will help you and facilitate you getting trained or getting the necessary training that you would like with this software is to pair this up with a mentoring program. Again, my request is that some of you who have more expertise and have worked with R and have a little bit better understanding of the tool, if you can step up to take on being the mentors. We can create pods, smaller groups that we can work together. And these pods can work on doing these homework assignments. And the mentor of each pod, we can actually work with the mentor. So giving them a little bit more of hand holding, so the mentor can bring his understanding, his or her understanding to their pod that they will be working with. Depending on how many mentors we get, we will be distributing pods of 10 or less, preferably, and then we can take it from there. Again, this is just the day one of the workshop. We still have another follow-up, which will be happening exactly a week from today, during which time we will have all your questions answered. But in the meantime, while you're working on your assignments, we will have your mentors guide you through the process. And again, in the interest of time, we will have to, it's 11 a.m. Eastern right now. And all the questions which we have not been able to address, we will address them via email, and the follow-up communications with you will happen via email. Again, thank you all for being part of this workshop. We will be in touch with you, and we will continue our learning for the rest of the week, getting our expertise in RxODE. Thank you, Wenping, and thank you, ISOP, for hosting this workshop today.
You're welcome.
Enrico?
Yes.
So we can share the recordings of the workshop with all our attendees as soon as possible, and have these follow-up conversations via email.